In this video, we're going to continue to look at various transporters, and so now we're going to focus on the ABC transporters. ABC transporters, remember ATP binding cassette, will transport ions, sugars, amino acids, polar, nonpolar, basically a lot of stuff. One type of ABC transporter in particular, P-glycoprotein, this is overexpressed in cancer cells. And if you cannot get rid of the ABC transporter, P-glycoprotein, it's very hard to kill cancer cells. That's because this transporter will pump out amphiphilic substances. Well, cancer drugs, or anti-cancer drugs, are usually amphiphilic substances. So cancer cells overproduce these. So if you try to give a cancer cell a drug to kill it, it's just going to pump it right back out again. The way that this works, the way how a lot of ABC transporters work, is that you have two nucleotide binding domains. This is what's going to bind your ATP. Remember, ATP is made out of a nucleotide. In this binding, you can get a conformational change, what helps pump out your substance. And in humans, you have 48 different ABC transporters that we know of so far. So, general way in which this works, so yellow here is ATP, is that a molecule, lipid soluble molecule, can enter P-glycoprotein through a little portal that's in its membrane spanning domain. You'll bind via these hydrophobic or aromatic residues. Once bound, you have ATP hydrolysis, which has a giant change in conformation. You show this molecule at the outside of the cell and kind of kick it out. So imagine that this is a drug. The drug is trying to go inside your cell, binds to this ABC transporter, drugs goes right back out. ATP hydrolysis can restore this channel. So binding makes the channel change. Hydrolysis makes it go back to this stage. Now, if we look at one type of disease, cystic fibrosis, this is actually due to an ABC transporter that doesn't work. Now, cystic fibrosis transmembrane conductance regulator, or CFTR, this is the only ABC transporter in humans that we know that actually is an ion channel. And this ABC transporter doesn't work for individuals with cystic fibrosis. Now, normally, this allows chlorine to flow out of the cell. But for 70% of cystic fibrosis cases, one amino acid, phenylalanine 508, has been deleted, and this channel no longer works. So you don't have as much calcium flowing into your airways, which means that you will get a thickened mucus. You cannot clear this mucus from your airways, so you're constantly coughing. You get infections a lot easier. You have a hard time breathing. There's a lot of different ailments that come from one amino acid being deleted in one protein. So let's talk about some more ways of transporting. So we're going to go back to glucose transport. And I talked about in a previous video how the sodium potassium ATPase helps to generate this electrochemical gradient across your cell to make the inside negative. Well, we can actually use this to our advantage, so here's the ATPase, to help transport other substances, one of those being glucose. So this is what's called secondary active transport. We're using the sodium potassium pump, which is ATP, we're coupling that to put glucose in. So the idea is that you have high amounts of sodium on the outside of your cell, small amounts of sodium inside. So it's favorable for sodium to go inside your cell. We use that favorable energy to help bring glucose inside the cell as well. Then glucose can continue on and it will want to leave the cell, and it can do that via glucose uniport, GLUT1. Then we need to rebalance our sodium, so we'll kick sodium out, bring potassium in through our sodium potassium pump that uses ATP, that is secondary active transport. So it's technically active transport, but we're actively transporting sodium and potassium, even though what we really care about is about glucose. And so this actually happens in your small intestines. This is how we take glucose and just get it through that intestine wall and into the bloodstream. And interestingly enough, this transport of sodium actually makes the cells uptake more water. So if you have individuals who have severe water drought, I'm not sure what I meant by SAIL, but they have severe water loss and drought, like diarrhea, for example. The way to help them make better is not to give them straight water, but to give them glucose, salt, and water. Because of this pump, you're going to actually take water along with you, and you will absorb water a lot better than if you just had sugar and salt. Now, one more type of transportation system we're going to look at is lactose permease. Lactose permease is how we bring lactose into cells, and you are actually utilizing the proton gradient in bacteria to transport potassium and lactose. So the proton gradient, you might have heard of it before. It's from oxidative metabolism. We have a proton pump that will pump H's, or hydrogens, outside the cell, and this takes energy. So for potassium to go back inside the cell, that's more or less energetically free. Well, the lactose permease, again, has two different forms, E1 and E2. This is no different than the other types of transports we talked about. So E2 will bind this hydrogen to make E2 hydrogen. This E2 hydrogen form binds lactose, and you'll get this high energy intermediate. So it will relax the E1 conformation and release lactose. It will also release hydrogen, which can be pumped out again. And then in the E1 form, you have to go back to the E2 form, and then you can bind again. So this will only switch once hydrogen and lactose are bound. So we are making sure that we're not just bringing in hydrogen, because really we want to bring in lactose with that hydrogen. And our energy for this is really coming because we have high concentration of hydrogen here, low here. So bring in hydrogen, energetically good. We bring in lactose with that as well. That's it for that video. See you in the next one.